Right, good morning, everyone. And thank you very much to the organisers for inviting me. I really enjoyed the talks yesterday. It's not a field that I'm very familiar with. So I'm absorbing everything currently. So I hope today I can tell you a little bit sort of broadly about managing data resources as we do at the EBI, European Bioinformatics Institute. And I hope we can both learn from each other. So without further ado, briefly, but just to if people don't know the organization that I work for, it's an intergovernmental organization. This is EMBL, European Molecular Biology Laboratory. And it has run from 6 sites. And you're probably most familiar with the Heidelberg site because this is the home of Eurobiomaging, along with the Finnish leadership. And, you know, the EBI is based in the UK and it's the second largest site of EMBL. And our focus is bioinformatics, and it's probably fair to say, well, we mirror all of the goals of EMBL in general. We sort of lead with our data services. We have research, we have training, we have industry links, and we coordinate bioinformatics in Europe via the Elixir infrastructure. But our bioinformatics resources are world leading and world famous. So the EBI data resources are used relentlessly every day, 24/7 from all over the world. Basically, we receive about 100 million requests for our data services every day on our websites. And there's about, in a given year, there'll be sort of 40 million IP addresses, unique IP addresses that access the resources. And, you know, really, every 10 minutes, a paper is published that cites an EBI data resource. So it's really, I think, even though we're called the European Bioinformatics Institute, we've really gone beyond that now, I think, to become fundamental for any life science research. There's probably people using the data resources without really realizing they come from the EBI, but this is data is gone beyond bioinformatics these days. And so the resources that we run cover the whole of life sciences, from small molecules and chemicals, metabolites, genes and genomes. Of course, the European Nucleotide Archive is one of the oldest archives in the world, full of sequence data. But we also have genome data and RNA data as well. Then the world of proteins, we run the PDB in Europe, the PRIDE database of proteomics, and UniProt database of knowledge on all the different proteins. And of course, imaging and cell structure, which you're probably more familiar with than the rest of it, which is we more recently launched the Bioimage Archive and the electron microscopy database, data bank, and the EMDB also. Sorry, I'm just going to move this. Then in terms of genetic variation disease, we have a Decipher database and some databases that relate to human controlled access data in terms of the Genome Phenome Archive. And then, of course, all the databases are no good unless you know the biological context of the resources. So this includes the literature databases, which I have run historically, but no longer do, and ontologies and pathway databases and so on. And there's 2 things that we have a lot of faculty. There's about 500 people who run all these data. It's quite an enterprise. You know, 500 people run these resources. And 2 of the things we are constantly thinking about is how can we sustain these resources and where are the scientific horizons? Because when you run these resources, you have to really be thinking about, you know, where next and how can we anticipate that need. Another point I like to make here is that these resources, you see the sort of little orange dots and the blue dots, and this means that those resources have been recognized by both Elixir and a newer organization called the Global Biodata Coalition as fundamental resources that support life science research. And they come in 2 forms. One is submission databases. So this is when the communities that use and develop and generate those data submit the data to the database, and another type of database where we build on top of that called an added value database. And a good example of that is the European Nucleotide Archive, which takes submissions from all over the world in collaboration with other nodes of the INSDC. And then on top of that, we build genome builds and annotate those genomes in the Ensemble genome browser. So thinking about sustainability, and sort of there's many ways you can think about sustainability. And one of them is sort of a technical approach. Of course, there's funding, which is very important, as well as partnership and scaling issues. So here, this is the yearly volume of deposited data in these resources, and it's a logarithmic scale. Otherwise, it wouldn't be very useful to look at. Otherwise, we wouldn't get it all on the same graph. And at the top, you see the European Nucleotide Archive and the European Genome Phenome Archive. And these are the biggest databases in terms of the volume of data. And about 90% of all the data we have at the EBI is sequence data. But if you look at the green line, which is EMBL-EBI, the electron microscopy database, now that quite recently overtook the proteomics in terms of size. And you can also see the bioimage archive there in blue, also on the, with the steepest slope of increase. So basically, while the sequence data is the biggest and will remain the biggest for some time, the imaging data is the fastest growing. And while at the moment it's orders of magnitude different, total researchers deposited about 100 petabytes in the last year. And at the moment, 90 petabytes is DNA, but the imaging is coming up. So I think, if you projected that into the future, there might be a day in the not too distant future where the imaging data overtakes the sequence data. We'll see. But this is the kind of thing we have to anticipate and plan for. So it's really important. And another sort of route to sustainability is that we can't hold all the data from all over the world and do all of it. So it's really important to do this with partners. And one of the things about our delivery partners is that they're very long-standing, and they basically have institutional backing to do the kinds of things they do. It's very difficult to do these things just for, you can't do it for 3 or 5 years. You have to be there for the long haul. So there are various ways of doing this, and various ways of sort of almost federating the data and federating the responsibilities. The oldest database in the world is the Protein Data Bank, that's been doing this for over 50 years now. And there are a number of nodes of the Protein Data Bank, including now China and India. So it's going more global than it used to be. And the way they do it here is that there's all these different websites, but they all point to exactly the same FTP site held in the USA. Another good example is the INSD. This is the International Nucleotide Sequence Database Collaboration with partners in the USA, Japan, here DDBJ. We'll hear from Matsunori tomorrow, I think he's going to be here. And the European Nucleotide Archive in Europe. And this is over 30 years old, and actually has just launched a call for more global participation, acknowledging that this whole enterprise needs to be far more geographically spread across the world. In this case, the federation is that each node can take submissions, and then the data is moved around the world. It's shared with each node on a daily basis. Now, this is becoming quite difficult to do because the data is getting so big. When the sequence databases started back in the sort of 90s, it was still challenging then because our technology was so different, but it's becoming increasingly more difficult. And then finally, the proteome exchange. This is proteomics mass spec data. This data stays where it is, and the federation is on the metadata layer, which allows you to query across these different sites. So that's another model, because that data is pretty enormous. And then I think there's an extreme version of, this is the most extreme version of federation, and this is used for the Genome Phenome Archive, and this is because it relates to access human data, which means that data typically cannot move across national borders for legal reasons. And so here, this is a truly sort of, I would say, the most ultimate federated model where the data stays in the country. And again, the metadata layer allows you to query that data in safe ways so that you're not exposing the data until someone has filled in a controlled access data form and been given permission to access the data more fully. So that's much more controlled. And again, this is mostly in Europe, but is now becoming a more global approach to managing human controlled access research data. So different options available. Oh, my slide's gone a little bit wonky, but basically bioinformatics has the potential to bridge biological disciplines. When you take a computational approach to data analysis, you are then starting to look at different data types across different systems. And so this is one of the powers of having data in databases that understand the data, that have the right kinds of metadata, that mean you can query across them all and do some analysis. In fact, there's a bit of a joke about bioinformatics, which is that it's all about mapping identifiers, and there is quite a lot of that goes on. But this really can span from molecular biology, in molecular biology, it's structural biology, cellular biology, developmental biology, organismal biology, and going into different scales now, into ecology, evolution, and even beyond biology, into aligned disciplines. I mean, in particular in the era of climate change, understanding the impact of the environment on living organisms, whether it's human health or biodiversity or food security, are incredibly important areas of research. And so I'm just going to take a little bit of a forward look now and tell you a little bit about some of the things we're thinking about how we provision for the future to really continue to capitalize on the opportunities of computational biology. And some of this will involve imaging. I think we have this full suite of databases at the EBI that go right from the very old, the PDB, 50 years old, molecular modeling, atomic structure, very small scale, to potentially very large scale on a cellular level, light microscopy, and this is the Bioimage Archive. I've seen it on lots of slides actually yesterday, which was really great. I'm not going to talk about this now because Matthew Hartley, my colleague, is going to talk about Bioimage Archive specifically later. But including Empire and EMDB, we have the opportunity now to go across scales in multimodal imaging techniques, which are definitely being applied now to different biological questions. And I think we have the opportunity to develop the necessary infrastructure to support these amazing advances that we're seeing in science. I've heard this word spatial transcriptomics come up a couple of times, and again, this is a very exciting area of work. Historically, in trying to understand disease, the imaging has been purely morphological. So we look at structures through the microscope, we look at the pictures and understand what the disease profile looks like. More recently, we've had single cell transcriptomics is exploding. Everybody's doing it. There are a lot of different methods out there. There's a core method that seems to be becoming more standard. But this basically allows us to query that tissue and understand the different cell types that are within that tissue based on the translational profiling. So, and you have this kind of diagram, which I'm sure everyone's seen, where the cells are clustered according to similar types. And what the spatial transcriptomics promises to do is to link back those cell types to the morphology, the tissue structure. And this really then shows us more deeply, moving forward, there we are, where those specific cell types map onto the structure and therefore give us much deeper insight into what's happening in those tissues in disease states. Now, this all sounds fantastic, does it not? But how are we going to manage the data for these kinds of experiments? So, of course, we do have these databases that will take all these kinds of data. We now have the Bioimage Archive. We have the European Nucleotide Archive for the RNA-seq data. And we also have databases for mass spec, protein, and metabolites, actually, for the metabolomics. And you can see, very nice papers that do all of these things and come up with, wonderful pictures in the paper and wonderful, interesting conclusions. But at some point, we will need to store all this data in a way that can be reused. And it's really not that trivial. If you think about it from the perspective of the poor submitter, are they going to have to submit potentially 4 different types of data to different databases to comply with their funder and the journal requirements? And if they do that, how are we supposed to know that they are all linked? So I think, even just the very basic things like linking the sequence data in one database to imaging data in the Bioimage Archive is actually not so straightforward, let alone coming up with browsers that are easy for people to use to, to look at these maps and interrogate them in the future. So this is one thing we're thinking about. Another thing that we do, which is another sort of sustainability model, is to use data portals. So rather than invent a new database for every different data type, we put all the data in the fundamental database, like I just mentioned, for spatial transcriptomics, and then build these portals on top. And the one that perhaps people have come across relatively recently is the COVID-19 data portal. And this really pulls again all kinds of information from all kinds of core data resources. Similarly, the biodiversity portal, there's also one for the farmed animals community in Europe, which really allows the archival data to be held in the most appropriate way, as well as the community being able to add back any annotation or other information that they like. For some reason that just appeared, but there's also a pathogens portal. Okay, so we've got our communities with the portals. We've got the challenges of multimodal, multi-scale data to manage. But if we can do this all well, this means that we will have even greater opportunities in the future to make tools that analyze that data for us, because the most powerful querying will come from being able to query across all data types in a very easy way, in a natural language way, potentially. We're a long way off doing that yet, but there are some things that we definitely need to do to be able to support those kind of AI approaches. And we do many of these things right now, but they're going to have to be expanded. And this comes to coming back to some of the things that was being said yesterday about managing data right off the facilities, capturing the right kind of metadata as early as possible will make all of this kind of thing easier and more straightforward in the future. So firstly, data standards is absolutely bread and butter work for some of our data resources at the EBI. Often we have a convening role because of the running the database on behalf of the community, but it's the community that decides these standards. Nobody dictates them. So we're going to have new ones to support the increasing size and complexity of data. There are definitely missing ones that would be really valuable to have that we don't quite have yet. And then furthermore, adding the right kinds of ontologies to annotate the data with will increase the ability to interoperate that data on a semantic level. We're going to need to scale our curation efforts. So one of the things that all these AI engines need, of course, is high quality annotated data. And the more, the better. So our curators are never going to be out of a job, but their jobs might shift a little bit because we do need this expert manual or I don't know what the gender neutral version of manual is, but manual curation is absolutely essential. And people can still do things that in complex contexts that machines cannot do. And then when we've used that, then we can reuse those expert manual curations to make better AI models to help us with the annotation process itself. There's a lot of talk about farification. I think we do fair data at the EBI. All the data is fair. I think there's degrees of fair, and people seem to think about fair in quite different ways. But fundamentally, all the data at the EBI can be cited, and it also can be reused. Now, one could but one can always do better. I don't think there's any ever sort of definition, working definition of fair. So we need to make it more machine readable. What do the machines want? I mean, we used to get serving up data to biologists, not so much machines. We're going to have to, I think there might be programs in which we create bigger and powerful reference data sets in order for them to be then fed into the machines. And I think, sort of projects not dissimilar to the Human Genome Project, perhaps not on the same scale, but definitely projects where we're injecting that kind of missing data into the systems to allow us to do the AI development. And then also thinking about, we traditionally serve up data from different resources, and you have to go sort of like shopping to the different places to get what you need. But actually, in the future, we're probably going to have to deliver data in a sort of more dynamic way across data types. For example, I mean, a simple way to think about it is to think about if you were interested in all the data that we had on fungi, you know, can you get that in an easy way just with one query to pull it all back for your AI development? And then finally, this community engagement that we do as a key part of running these services to, we got to bring it to new audiences, and also when people make models, we're going to have to we can help with the benchmarking, because the data will not only generate the models, but also benchmark. So scaling up is something that we think about in terms of the annotation, as well as terms of actually just hosting the data. And we get these projects, a bit like driver projects, that really push things in a certain direction and sort of keep us like a little bit uncomfortable, let's say, in trying to manage what science and research is going to come up with next. And in the area of genomics, biodiversity is a huge push to make us scale up our efforts. And we've basically got about a few hundred eukaryotic genomes in Ensemble at the moment, assembled and annotated. And this is going to go up to hundreds of thousands with all these global biodiversity sequencing projects. But I do think it's not beyond the bounds of possibility that within a few years, we will have good AI that will be able to do the gene calling, that is the annotation of the genome sequences. Why? Because the data sets that we have are big enough and rich enough and accurate enough through the manual annotation to be able to train such engines to do this gene calling. And this will be particularly important in biodiversity projects, because there you have less expression data to do the annotation in areas of the phylogenetic tree that have been are less well studied. So this will be essential to ramp up the efforts of the annotation this way. So that's what I would predict happening relatively soon, I hope. And then it's hard to talk about the EBI data resources and the PDB without mentioning AlphaFold. And this is just an inspirational project. Is really transformative to not just structural biology, but everything that comes from structural biology, all of the subdisciplines and all those that use structures in their analyses. And this, of course, just won the Nobel Prize. So you probably knew that already, the Nobel Prize for Chemistry, for Demis Hassabis, John Jumper, and David Baker. And it's really, it's been transformational, and it's not been out very long. There's already 12,000 papers that are citing the AlphaFold algorithm or the AlphaFold database. So what we did was, because they had used all the open data in the EBI databases to train the models, we then collaborated to make the AlphaFold DB, AlphaFold database. So this used the algorithm to predict structures for every known protein, every protein that we know about, which is every translation from the ENA that's in UniProts. That's over 200 million proteins. So if you think about the PDB, it took 50 years. And as a colleague of mine in structural biology said, the blood, sweat, and tears of many graduate students and postdocs over half a century to build this data resource that then fed into, which has about, I think it's a couple of hundred structures. So from a couple of hundred experimentally derived structures to 200 million in a matter of months. And this is why it's transformational. And I hope that we see many more advances on the lines of AlphaFold in the future in genomics, and yes, maybe also in bioimaging as well, ultimately. So with that, I'll finish, and I'd like to thank all of our collaborators. This is none of this is possible without a lot of community engagement, and this is just this is most of the groups that we work with at the EBI, and they all have different roles. Some are projects, really sort of cutting edge science. Some are infrastructure partners, who we've worked with for a very long time to produce the infrastructure. Others are standards development, community outreach, and capacity building. And it's really not, and you probably see some familiar logos, I hope, on that slide. So it remains for me just to thank all of our staff. They are over 800 strong, very international, and they're very multidisciplinary. It's not all research. These are data scientists and data stewards, bioinformaticians, a lot of software developers, as well as subject-specific subject knowledge people, especially our curators. So with that, and our IT department, incredibly important. So I would like to finish there, and thank you very much.